So hello, everyone, and thank you for joining in for this talk entitled Programmable Genomic Integration, a technology that enables the integration of large DNA sequences at specific genomic locations, presented by Dr. Sandeep Kumar, Senior Director, Platform at Tome Biosciences. For a complete biography of today's speaker, click their name in the presenter window on your screen and then on the biography link. I'm Sean Taylor, and I'll be your moderator for today's event. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Q&A box. We'll answer as many questions as we have time for at the end of this presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Dr. Kumar, you may now begin your presentation.
Hello, everyone. This is Sandeep Kumar from Tome Biosciences. So today I'll be talking about the work that my team or the entire Tome team has been doing for past few years. At Tome, we are working on programmable genomic integration. But before I go or do deep dive into the content, I want to show my disclosures. So I'm an employee and a shareholder of Tome Biosciences. So before I go into the technology piece, I wanted to talk about, spend a little bit of time about PGI. What do we mean by PGI? So with PGI, it is a programmable genomic integration. And in this overall concept, what I'm trying to get to the point is that with this technology, we are able to integrate or insert any piece of cargo anywhere in the genome. Now, I understand that this is a bold statement. Until now, technologies for programmatically inserting the code anywhere in the genome have shown limited success. But with this, when the defective copy is replaced by a functional copy and is still under the control of its core promoter and the distal and the proximal regulatory elements, there we assume that there will be a limited amount of supraphysiological expression that comes off from these. So we believe that this will be the best way or the way to edit the genome. And the next thing when I say the PGI or the Programmable Genomic Integration, it is, as the P stands for, is programmable. So when I say it's programmable, that means I'm not talking about random genomic integrations, and I'm not talking about integrations somewhere in the safe harbor site. What I'm talking about is any user-defined location on the genome. So you point a finger that this is where I want to go, and with this technology, we'll be able to get or insert a big piece of cargo or DNA or any sequence in that position or in that place. Directional. So by directional, what I mean is that every insert that goes into the genome is in the same orientation. It's not flipped backwards, so the final and edited product is going to look exactly identical. Efficient. So when I talk about the efficiencies, hex cells are a good model organisms to start with, but at Tome, we in just very couple of early months, we were already reaching 80 to 90% of the editing efficiencies or the I-PGI levels. But here I'm going to talk about, but the hex cells are a good model to get started, but finally, the actual medicine has to happen or has to be made in the non-dividing cells or the cells that we care about, the primary cells. So the efficiency when we change from the hex cells to the more model systems, or which are true representation like primary human hepatocytes. When we moved from HEC to PHH, the efficiencies we saw, there was a significant amount of decline from 80 to 90% to below 5%. And then we did all the optimizations over there to bring it back to the current level. Any size. And when I say any size, with our technologies, we are able to insert from 1 to a couple of thousand base pairs. So far, we have shown that we can insert up to 31 kilobases of the code. But really, the caveat here is that if you can get that DNA into the nucleus, you will be able to insert that thing in defined position. So the delivery is the only bottleneck here. If you can get the DNA, this technology is going to insert it. And the next 2 points are like the multiplex and the low risk, is that since our technology doesn't rely on generating double-strand breaks, so it's not dependent on homolog or the HR pathways, we can multiplex this thing. So we can insert multiple codes at multiple different positions in the genome. And while jury is still out there, whether generation of double-strand breaks are safe or not, but I will say if we can minimize the generation of these double-strand breaks, we should. And since it doesn't generate breaks, so it is relatively low risk. With this, so we have several tools in our toolbox to accomplish these tasks. I'm going to focus today on 2 tools. One is the I-PGI or ligase-mediated Programmable Genomic Integration, and the second one is I-PGI or Integrase-mediated Programmable Genomic Integration. So what I mean when I say I-PGI, this is a relatively newer tool. In here, the programmable piece or programmability comes from the Cas nickase, and the code or the thing is written off by ligase. So here we are not synthesizing the code in the cell. We use a pre-synthesized code that we can ligate directly. So there is minimal DNA synthesis involved. So again, and then since we are directly ligating it, so there is a possibility that we can just have one lipid nanoparticle formulation. We have tested this thing. This can do 1 to a few hundred base pairs. We are still exploring it. This can be used for exon sites replacements. We do not know the upper limit on the system yet. And the second one, the I-PGI or Integrase-mediated PGI, is the real bread and butter. Like this is a real workhorse right now at Tome. And this technology doesn't care about the size, so we can deliver multiple kilobases of DNA or integrate multiple kilobases of DNA into the genome. And this is where we can think about logic gates or multiple cards, or it can be anything. The code can be anything. So a little bit more into the I-PGI is we look at this process of I-PGI as a 2-step process. The first step is we say at Tome as a beacon placement, but really that the first step is where programmability is coming from Cas9 nickase, and then the short code that we write is about 40 base pairs. It can be any attachment site, which is written by either a writing enzyme. This writing enzyme could be anything, a polymerase or a RT, or it can also be a ligase where we just directly ligate the piece of DNA into the genome. So that's the step 1. So when we write in a small code around 40 base pairs, we call that code as a beacon. Now in the step 2, this beacon is recognized by an integrase, our proprietary integrase. So the integrase knows where we have placed the beacon. So in step 1, we tell this is where we want to go, and we place in a tag or a beacon. And then in the step 2, the integrase doesn't care about the size of the DNA template. Integrase does what is supposed to do, or that's what it is evolved to do, is insert the big piece of DNA, which again has a small attachment site on it, and insert it wherever we place the beacon. So this is how the overall PGI technology works. Now at Tome, we are developing this or evolving this technology for 2 different applications. One for cell therapy applications, and then for the in vivo applications. For cell therapy is a different mindset in design, and for integrative genomic therapy is a different set of mindset. I'll go over each of them. But for cell therapy, again, we are thinking about we can multiplex it. We can generate these different iterations very fast. And what we realized very early on that now for the ex vivo applications, that we have to stay focused. We cannot move in multiple directions. And this, so as our CSO and CEO said that there are multiple avenues that we can explore, but if we stay focused, then we can reach the end goal faster. So here we decided that we will be focusing on iPSCs, and the only reason being that these iPSCs can then be differentiated into any cell type as long as we have good efficiencies in there. So we decided to work on iPSCs. And as you can see on the graph on to the left is the efficiencies. This is very early data in here that we were getting good efficiencies of putting a cargo at a defined location or a code in a defined location in the genome. And the slide in the middle shows that we can go as high as 31 kilobases. Really, the limit was how much we can deliver into the cell. And this 31 kilobases was brought into that cell by adenovirus. But again, Integrase doesn't care of the size. And then the slide onto the graph onto the right shows that we can multiplex it. Again, this is one single setting where we placed beacons or attachment sites at 4 different places, as well as, so this is a one-step electroporation, and the final product is the I-PGI. So you can see that it works with varying efficiencies in all 4, but we can see insertions in all 4 positions in the genome. With this, so what do we do if we can engineer the iPSC? So what we can do at home? So we thought about it for a long time, and then we decided that we will deploy this to autoimmune diseases, and we're going to have this iPSCs differentiate to iNKs. And that's our first, the lead program as well. Here we are trying to do multiple edits onto this iPSC, and then so we spend a lot of time to design what should be the code, because we can insert up to 12 kilobases of code into the cell. Not in this situation we have inserted 12, but as I said, 31 is really the upper limit. Is that it has the efficacy edits like CD19 and BCMA dual CARs or single CARs. We have engineered in the safety switch so that if the cells go out of hand, we should be able to turn the cells or kill the cells off from the system. We have done some edits so that in future, if you have to do antibody-mediated lymphodepletion, we should be able to do that. It has some cell fitness edits, and it also has the allo shield so that they are invisible to the endogenous host defense mechanism. And so now once if we have generated a cell like this, how does the efficiency looks in the cell? So we, as a first program, we are inserting in 12 kilobases of the code, and with all the edits that I talked about. So if we look at the expression onto the left, both the BCMA and the CD19, you can see we can observe around 20% expression for both. And if you look at the graph in the middle about the allo and the iNK fitness genes, they also kind of express at the similar levels. So we are able to generate an iPSC, and all the cargo or all the code that we placed in the cell expresses. And in a separate experiment, we also confirmed that all these things not only express, they also function as they are supposed to function. So onto the graph on the top top half, you can see where we looked at whether the safety switch that we have engineered in, how it's working. So we can see that when we add the small molecule activator, that safety switch is able to, that small molecule activator is able to kill the cells. And the graph on the bottom is for the fitness, and I'm not going to go into details, but you can see based on the cells who have these fitness genes in this, they don't need the cytokines provided from the outside. With this, I'm going to show another last piece of data in here for the ex vivo application. Is so we asked a question, okay, these individuals are working, so how are they able to kill the cell? So here we use SLE patient PBMCs. So SLE is those who don't know is also known as lupus. So we took the cells from the patients, and we differentiated our iPSCs to iNKs, and then we asked the question, are they able to kill those B cells? And as you can see on the graph on the right, that at various effector to target ratios, these iNK CARs or the CAR iNK cells were able to eradicate those B cells in a CAR-dependent fashion. So this was the last piece of data, and now I'm going to switch gears, and I'm going to talk more about the in vivo applications. So in vivo applications, we are trying to go more simple. It's not like complicated cars or any of those things. This is more simple. Here, the premise is that we will just fix in a broken gene. So we will put in a functional copy of the gene right in front of the defective gene. And to do this, as you can see there, what I'm showing here is that we need the lipid nanoparticles as well as the viruses to take the cargo in. So our system, what our founder says, is sophisticated. It's not complicated. It's sophisticated. So we try to stack our risks. So there are already lipid nanoparticles that exist which can take the cargo or the editing machinery easily into the liver cells. So we are using lipid nanoparticles for the editing enzymes. And the AAV particles are the ones which can take in the code or the DNA into the cell, into the liver cell. So that's what is the delivery. And we are trying to put in the functional copy of the gene in our intron 1. So as you can see that, so the defective gene, we put the functional copy right in front of it so that it expresses seamlessly. With this, a few data slides, because now how do we do that? That's where it took me a while to think about what data I should be showing in here. What the data shown here is onto the left is this is the data coming from primary human hepatocytes. I-PGI in those. We tried different locations, different sites, and we were getting reasonably good efficiencies in any loci. I don't think that so far we have found any location in the genome where we were not able to edit it. So then we, as our first lead, we decided to focus more on the PAH locus because we wanted to work on PKU disease. And as you can see on the graphs on the right, then we started optimizing this system to increase the efficiencies further. And as you can see, over time, we were able to improve the efficiencies, and we are now in a very good efficiency level. And then when this cargo is inserted into the exon 1, now, because we are not relying on the exogenous promoter, that's one thing that I forgot to mention earlier. Our cargo is just plain the splice sites and the open reading frame. So the graph onto the right shows that not only we are able to place that cargo in the right place, it also expresses. So the splicing and all those things are happening seamlessly. We were very excited with these results, and then we decided to move on to the rodents or the mouse, and that's where we observed unexpected biology. So again, we use the lipid nanoparticles to place do the first step, or the beacon placement. That's what we were trying to get in there. We were able to get into almost every liver cell in the mouse, but what we observed was that only less than 1% of the beacons were usable. That means the majority of the beacons that we were placing in there, they were having truncations or writing something different. And so this is unexpected biology. And then we asked, wait a minute, what happened here? So is this a mouse problem, or this is a mouse cell problem? So next experiment that we did was that we evaluated whether it's because when we are looking at the mouse, it is the liver cell, which is non-dividing. So then we use dividing mouse cells, which are HEPA-16, or the non-dividing hepatocytes, PMH. And as you can see on the graph onto the left, that the fidelity is fantastic if you're using the dividing mouse cells. So really the problem is the non-dividing cells in the mouse. And we tried a few mouse strains as well, and every mouse strain we were not successful in writing in the correct code or the correct beacon. But gladly, this is a rodent-specific problem. As you can see on the graph onto the right, it works well in primary human hepatocytes. It works, the fidelity is fantastic in the primary cynohepatocytes. So it's really the mouse, and we are not working to cure the mouse diseases. So we decided that it's a rodent-specific problem, and then we decided to move forward and trying to solve it in the clinically relevant model. Then this was the whole reason why we started to explore the relatively newer technology, the ligase-mediated PGI. And as you can see over here, I know that I'm running low on time. As you can see over here, we are directly able to ligate a pre-synthesized piece of code. The fidelity looks fantastic. You can see this is the data from primary mouse hepatocytes. So you can see that from less than 1%, we were with just one or two initial experiments, we were close to 40% beacon fidelity. And this not only works well in the primary mouse hepatocytes, it also works well in primary human hepatocytes. That's shown onto the graph onto the left. And it not only works with equal efficiencies compared to the I-PGI, it also works, the fidelity is also slightly better compared to the previous version. And then we decided to focus on the second piece of the technology, which was the insertion of the code. So how we got around that was that we made some mouse strains where we are using pre-synthesized at B site placed into the mouse genome so that we can work on the second part of the process, which is the insertion. And as you can see, when we started our generation 1 molecules, we were getting very low efficiency, but over time, we were able to improve this efficiencies by about 25-fold. And as you can see in the mouse, we are easily able to get into the every mouse cell. And as we were doing these experiments, we realized that, wait a minute, so so this is the bulk alleles and how many cells we are reaching. So we did the immunostaining for, before going into that, as a tool cargo, we were using factor 9. So here we are putting in the human factor 9 into the mouse factor 9 location. And when we did the immunostaining, then to our surprise, we noticed that instead of cells looking like they are full of those, the human factor 9, they were excreting the factor 9 into the interstitial space, and which looks exactly identical to how it looks like in a normal human liver. So the gene just not only, the liver looks perfectly normal. That's what I'm trying to get to. And when we counted these number of cells, it looks like we maxed out. This is the very good efficiencies. And then the final question was, does it work in non-human primates? So this is the last data slide about non-human primates. So here again, I'm putting in. I'm going in either into factor 9 locus or in the PK or in the PAH locus, and I'm putting in our tool cargo, which is factor 9. And as you can see, as with different experiments done, we kept on increasing our efficiencies. We are not nowhere done with increasing these efficiencies. So this is just the beginning. This is not the ceiling, and we believe that we will be able to improve these efficiencies further. But we, as a get-go, we are around a 10% bulk liver alleles that we can edit. And these not only get inserted, they also express, which is shown onto the graph onto the right. Now I'm gonna just spend some time onto our lead indications that we are focusing at Tome. So we are on the in vivo side. We are not only focusing on just the rare diseases. We are also, as you can see in our pipeline, there are some common diseases as well, like ASVCD, and on the cell therapy side, about the autoimmune, where there's a big population out there. With this, I'll end the talk. But again, this is not my story. This is the whole team. It's a good teamwork. And I would like to thank our CEO, Rahul Kakkar, and our CSO, John, and our founders, Omar and Jonathan. And with this, thank you. I'll take questions from the audience now.
So thank you, Dr. Kumar, for your informative presentation. That was very fascinating. It looks like with PCR, we have been able to find the proverbial needle in the haystack. And now with PGI, we can place that needle back in the haystack very precisely. We're going to start the live Q&A portion of the webinar now. If you have questions you'd like to ask, please do so now. We'll answer as many of the questions as we have time for. And let's get started with the first question. So the first question is from the audience. And the question is, what are your future plans to increase the efficiency of the process?
Yeah, as you can see, it's a really good question. As you can see over here, there are multiple, what we say in Tome, there are multiple knobs, and you can turn each of these knobs to get the efficiencies higher. So as a I can break it down to two buckets. One bucket will be improving the enzymes. We are still working on how we can improve both the writing enzyme as well as the integrase enzymes. And then those will be the ones where enzymes are overactive, or maybe I'll say working at the peak activity. Then we can also look for how we can increase the expression of these enzymes. So those comes from the messenger RNA improvements. And then we can also then work on the lipid nanoparticle formulations, because this drug, as I said earlier, is a little sophisticated. So we need lipid nanoparticles and we need the AAVs. So we can improve the lipid nanoparticles. We can then work on the dosing timings. And those are the not all of the exhaustive list. These are the kind of factors that we are playing around currently to see what is the right dose and the right amount and the right enzyme that will get us to the peak levels.
Okay, kind of a related question on the subject in general, Dr. Kumar, is again one from the audience about has this technology PGI been published? Where can they learn more about the technology on their own?
This PGI technology is an extension of the paper that our founders published on PASTE. So I will say that this PASTE manuscript was in bioRxiv and then later on published in Nature. So I will refer users to refer back to the PASTE from Omar and Jonathan, and Omar Abudaya and Jonathan Gutenberg. So that will be the place where you can learn more about this technology, the I-PGI. The L-PGI, I will say stay tuned. We are going to have a little bit more coming out in the ESGCT, which is in, I think, next month. And the paper, we are planning to have it submitted soon. So in a couple of months from here, we should have the L-PGI piece published.
Awesome, great news. Okay, a more technical question. Which vector should we consider if we use more than 10 kb, given the carrying capacity of AAV is less than 10 kb?
So yeah, I think as I said earlier, the caveat is how much code, like this technology doesn't care. So the AAV is the one which I will say is currently can get you to the liver. So if you are, say, working in the in vivo field, then AAV is right now is the only vector, but there are some other vectors that people are working on, but those are not, I will say, clinically de-risked. So the clinically de-risked vector as of now is AAV, and the max code that can be delivered is going to be 4.7 or somewhere around, I'll say 5 kb-ish. That is in vivo, but if you look ex vivo, then the choices are limitless. I'll say you can, the 10 kb or 12 kb code can easily, I mean, these are plasmids. You can use either the standard plasmids, or you can use nanoplasmids or mini circles or any other plasmid system. Because 31 kb then again gets challenging to deliver with the electroporation, and that's when we just had the adenovirus in our lab and we used it.
Okay. Another question, interesting question actually, about chromatin, the chromatin folded status. So how does the chromatin status of the target locus affect insertion? Are some of the areas of the genome that are functionally inaccessible due to interference from compacted chromatin?
Yeah, that's a good question. Actually, that's why, so our technology, because we go into the intron 1, So it's a whole big region which we can scan. And so basically in our technology, we just screen for optimal guide pair. So we do see that there are some regions which are more accessible and some which are less accessible. So that's why our first piece of the technology is that we screen for the whole region and see where we can put this code. But this is not it. Then we have to see not only this code is being, or say, inserted there, is it going to generate the functional protein? So we have to do the downstream looking at the splicing has to be not impacted by that, and any other factors which can interfere with the protein expression. So there are a couple of things that we have to keep in mind. But yes, you're right. The chromatin accessibility will be there. Issue will be there.
Okay, fair enough. And then another technical question is, what is the background insertion rate in the absence of a targeting beacon? I'm guessing this is more like off-target type of situation.
Yeah, that's a good question. So the integrase that we use, it doesn't have any naturally existing sites in the human genome. So this is why we say like the on target. So we place the beacon, and that's the target we are placing in. But I think recently, David Liu also in the ASGCT presentation, they shared a little bit more insights on that. And then in our group in-house, we can see there are some off-target sites which we say as off-beacon, but those efficiencies are fairly low. But again, then we are engineering these enzymes not only for increasing activity, but for increased specificity as well. So our enzymes, we have reduced the percentage of any off-target insertions that happen in the naturally, or I'll say the cryptic beacons.
Okay. So then what happens to the leftover at B landing site or beacon in the exon 1 if no integration happens?
Yeah, that's a good question. So because we are in the intron, so that's not a part of the functional open reading frame. So if something is left back in there, it is not going to interfere with the actual splicing process. And besides, the gene downstream is anyways broken. So how bad it can be, like broken gene is anyways not functional. So we assume that it is not going to generate any aberrant product because it's just an inert piece of 40 base pairs sitting in there.
Okay, that makes sense. Since the drug product is quite sophisticated, do you envision any manufacturing challenges like downstream for commercialization?
Yeah, our technology is two lipid nanoparticles and one AAV. So we realized very early on that we have to have teams working on each of these aspects. So from very early on, we had the CMC teams working very closely with the research team. So now we have teams like the CMC teams working on how we can manufacture AAV particles, how we can have these long ATG RNA synthesized, how we can make these lipid nanoparticles in-house. So we have teams working in the message RNA, of course. So there are teams working on each of these, and we have a very strong analytical team which is developing assays to, I will say, qualify or say that these are very good molecules that we have generated in-house. So it's a team effort, and we started very early to focus on those CMC challenges. So now we are at the position where we see that we can marry all of those things together.
Fantastic. That's good news for the disease indications that you're working on. We have time for one last question, and it's a very general question, Dr. Kumar. So take whatever direction you want to elaborate. What do you envision as the future direction for this technology and for where Tome Bio is going with this?
Yeah, I will from here. So first and foremost, the major bottleneck in the field was that how we can do a big DNA insertion in a defined location. And mostly the cells that we care about do not have a good homologous recombination in place. So how do we put in a big piece of code? And how I see is with this technology can be applied anywhere. It can be used for human diseases, like what I have shown in here. It can also be used for, let's say, building recombinant plants, or I will say, if you can imagine anything where you can put in a big piece of DNA, this technology will enable you. So this has, I will say, other than human applications, that can be applied to any other field. It doesn't matter. You think about plants or, like, say, animals for desired traits or anything. This is I will say, solve the basic fundamental problem of inserting a big piece of DNA in a user-defined position.
Very, very interesting, Dr. Kumar. I'm quite certain you're going to get a lot of email inquiries after this discussion, and in your future discussions at the conferences you're going to. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. And once again, a big thank you to you, Dr. Kumar, for joining us today. That was a very enlightening talk. So just to let everyone know, we will soon begin our next session. So stay tuned, and we'll be starting the next session shortly.
Okay. Thanks, Sean, and thanks everyone.
You're welcome.